Crop plant anatomy is the study of plant parts and their function. Crop plant anatomy is the first unit of study because one of the primary objectives of the course is to provide you with a basic crop production vocabulary. Knowledge of plant anatomy is essential in communication among farmers, entomologists, pathologists, weed scientists, or anyone involved in crop production. Farm management specialists, local seed and fertilizer dealers, herbicide specialists, extension agents, and consultants all need to communicate about factors affecting plant growth. In order to convey accurate information and make the right decisions, a knowledge of crop anatomy is essential. Remember, if you are going to understand the whys behind production practices, you must know the parts of the crop plant and understand the functions of these parts. The first item for study is the section entitled The Plant Cell. You should study the general features of a plant cell, be able to identify them, and describe their functions. Remember, we are only briefly reviewing the anatomy of the cell and are emphasizing only the major cell parts and their function. You should now stop the VCR, study the section on the plant cell in your text, and then return to the VCR for further tutoring. The cell wall is located on the outside of the cell. The plasma membrane, a semi-permeable membrane, is located immediately inside the cell wall. The nucleus is the large globular body containing most of the hereditary material of the cell. The long strand-like parts shown here make up the endoplasmic reticulum, and the small disc-shaped bodies on its surface are ribosomes. Both the ribosomes and endoplasmic reticulum are involved in protein synthesis. The cytoplasm is the liquid or gel-like material of the cell. The chloroplast, found in the cytoplasm, is a principal type of plastid that contains chlorophyll. The chloroplast is the site of photosynthesis in the plant cell. A vacuole, also found in the cytoplasm, is a storage region for cellular materials. The vacuole regulates water pressure within the cell and maintains the proper cell rigidity. A mitochondrion is a site of aerobic respiration, which is the main source of energy for cellular metabolism and growth. Dictyosomes appear as curved strands and are associated with cell wall formation. Now let's move on to the section on seed and seedling anatomy. After you study this section, you should be able to identify the parts of grass and legume seeds and seedlings and describe the functions of these parts. This is a very important section in crop anatomy. It covers plant parts that will be frequently used when estimating quality of seed and when discussing seedling growth and crop management practices. Since many field-related problems are linked to poor seed quality, an understanding of seedling emergence and crop performance is necessary. You should now stop the VCR, study the section on cereal caryopsis anatomy in your text, and then return to the VCR for further tutoring. Let's now discuss cereal caryopsis anatomy using wheat and corn caryopses. The caryopsis is the botanical name for the cereal kernel. A brush of hair is usually found on wheat kernels of many varieties, but may be absent or different among different kinds of wheat. Brush characteristics are often used to identify and differentiate between wheat varieties. The brush of hair is absent on the corn kernel. Instead, the top of the caryopsis has a dent in it. This dent is formed because the soft starch in the center of the kernel shrinks more in the drying process than the flinty hard starch along the edges. The pericarp is the outer tissue of the kernel and serves mainly as protection for the caryopsis. The pericarp is ovary tissue, which means that the kernel is a ripened ovary. This is in contrast to a true seed, which is a ripened ovule. The alurone layer includes the few cellular layers that surround the endosperm of the kernel. This layer can be either colorless or colored and secretes important enzymes for germination. Dent corn has both starchy and flinty endosperm. The starchy or soft endosperm is located on the interior, and the flinty or hard endosperm is usually located in the perimeter of the corn kernel. The wheat kernel also has endosperm that is often referred to as gluten endosperm. This gluten is a matrix of starch and protein that gives bread dough its stretching ability, allowing the dough to rise. Corn does not have the types of protein or gluten that causes the dough to be as stretchy as wheat, rye, and triticale dough. The scutellum, often referred to as the cotyledon, is the embryo area of the kernel. Corn and wheat have only one cotyledon per seed, and therefore are called monocotyledonous plants. The coleoptile is leaf-like material that protects the embryonic leaves and shoots that make up the epicotyl. The apical meristem is also known as the growing point. After germination, future development of the stem and leaves will come from here. The scutellar node is where the scutellum is attached to the embryo axis. The axis is the line connecting the epicotyl with root tissue. The embryonic root found in the lower part of the embryo axis is called the radicle. Surrounding the radicle is a protective sheath known as the coleorhiza. During germination, the radicle elongates and breaks out of the kernel, leaving a rupture. The coleorhiza seals off this rupture to prevent disease from entering the kernel. The development of a black layer occurs in the corn caryopsis. The black layer is an abscission layer that prevents further growth of the kernel and indicates physiological maturity. A black layer is not noticeable in the wheat kernel. The final part is the tip, fastening the kernel to the plant. As you studied the figure showing corn and wheat kernels in your text, you should have noticed how similar the parts are for the caryopses of the cereal crops. Now we'll continue with the section on cereal seed germination and seedling establishment. Your study goal for this section is to be able to identify seed and seedling parts and know their functions during germination and emergence. A study of this anatomical information will give you a better background to troubleshoot field production problems occurring during the early stage of plant development. You should now stop the VCR and study the section on cereal seed germination and seedling establishment in your text. Then come back to the VCR for further tutoring. Let's look at the sequence of steps in germination and emergence of corn. Within the first 2 days after planting, the pericarp imbibes water, becoming soft and elastic. The coleorhiza elongates slightly and emerges from the pericarp. At 2 to 4 days after planting, the radicle then breaks through the coleorhiza and becomes anchored in the soil. 5 to 7 days after planting, the coleoptile elongates, and several lateral roots, called seminal roots, form at the kernel site. The radicle together with the seminal roots, are called the primary root system and is the main root system for the first 3 weeks of growth. The first internode of the stem, called the mesocotyl, elongates and continues until the coleoptile emerges from the soil surface 7 to 14 days after planting. When the coleoptile emerges, the mesocotyl ceases to elongate. As you study the wheat and corn germination and emergence sequences in your text, notice that the mesocotyl may elongate in some cereal species and may not in others. It elongates in corn and oats, but usually not in wheat, barley, or rye. This structure's elongation, plus the sword-like shape of the coleoptile, are some of the reasons why corn can be planted at greater depths. After the coleoptile emerges from the soil, the young leaves break through, turn green, and begin to photosynthesize. Coronal roots, also called crown or adventitious roots, develop immediately below the soil surface. These coronal roots are the secondary root system of grasses and become the principal absorbing roots after about 3 weeks of seedling growth. The growing point, or apical meristem, is located just above the uppermost node, also called the coleoptilar node. The growing point remains below the soil surface for approximately 30 days. It is this structure that indicates whether replanting is necessary after a cereal crop has been damaged by an adversity, such as hail or insects. The following steps will help you in learning how to dissect a corn seedling and locate the growing point. If the seedlings are big and have a lot of roots, you may want to prune them. Next, split the stem in half lengthwise using a razor blade. It is usually easier to start at the bottom or root end of the seedling. Open the stem and place it under the dissecting microscope. Look for the light-colored structure resembling a pyramid. At the tip of this structure is the growing point. Ask the instructor to help you if you have difficulty. We'll continue now with the section on legume seed anatomy. Be sure to study this section so that you know the parts of the legume seed and their functions. You should now stop the VCR, study the section on legume seed anatomy in your text, and then return to the VCR for further tutoring. Soybeans and alfalfa are two common legumes. Notice the vast difference in the pod shape and size, even though both pods are actually ovary tissues. The seed characteristics, however, are very similar. The radicle, hypocotyl, and epicotyl are the embryonic root, stem, and leaf tissues. Together they form the embryo area of the seed. The location of the embryo area, just inside the seed coat, makes it vulnerable to possible damage from rough handling during harvesting or processing. If the embryo area is damaged, the seedling will be abnormal or weakened and may not emerge after planting. The other parts of the seed are the micropyle, the hilum, the cotyledon, and the testa. Their functions are described in your text. The rubbery, elastic outer seed covering, called the testa, can be removed using your fingernail or a dissecting needle. At this point, the soybean seed can be split into 2 halves. Each half is a cotyledon, and therefore, legumes are called dicotyledonous plants. Look closely at each cotyledon. If care is taken when splitting the seed, the intact embryo will normally be attached to one of the cotyledons. The epicotyl, hypocotyl, and radicle can now be identified. The epicotyl is made up of the yellow leaves that will become the first true leaves of the plant after emergence. The epicotyl is attached to the hypocotyl at the cotyledonary node. The hypocotyl is stem tissue that elongates, pulling the cotyledons toward the soil surface during germination. In the area between the hypocotyl and radicle, the tissue changes from stem tissue into root tissue. At this point, the radicle begins. The radicle becomes the primary root of the plant. You should note that the growing point in legume seedlings, also called the apical bud, is located at the uppermost bud during the life of the plant. The location of the growing point within the seed is between the hypocotyl and epicotyl. Soybean seeds differ from corn seeds in several ways. Soybean seeds have a hypocotyl, but corn does not. Corn kernels have a coleoptile and coleorhiza, but soybean seeds do not. Additionally, the seed tissue does not contain part of the ovary, as in the pericarp of a corn kernel. The soybean seed contains 2 cotyledons, but corn has only 1 cotyledon, called the scutellum. Soybean seeds contain approximately 20% oil and 40% protein. Corn kernels contain approximately 60% carbohydrates, 4% oil, and 12% protein. The next section for study is entitled Legume Germination and Seedling Establishment. You should now stop the VCR, study this section in your text, and then return to the VCR for further tutoring. Let's look now at the germination sequence for soybeans. The testa allows water to be imbibed by the seed. The seed swells and becomes elastic. Hormones stimulate the growth of the embryo. The radicle elongates and ruptures the testa, and then elongates through the soil. The hypocotyl then begins to elongate, pulling the cotyledons toward the soil surface. The curved upper portion of the hypocotyl is called the hypocotyledonary arch, and it straightens when the seedling emerges. The testa usually falls off the cotyledons and remains in the soil. As the hypocotyl straightens, the cotyledons unfold, allowing sunlight to reach the young leaves of the epicotyl. The leaves turn green and begin to photosynthesize. The cotyledons will also turn green after receiving sunlight and photosynthesize for several days following emergence. The first vegetative leaves to appear after soybean emergence are unifoliolate, a leaf having a single leaflet. Then the trifoliolate leaves begin to form. They consist of 3 leaflets attached to a petiole. Swollen, knot-like nodules become visible on the legume roots. These nodules contain bacteria called rhizobium, which convert nitrogen to a form available for plant use. Axillary buds are present at each node above the soil surface. These buds can result in vegetative growth, such as branches, or reproductive growth, such as flowers. The apical bud, or growing point, is the topmost bud on the stem and is enclosed in the youngest developing leaf. New stem growth develops from the apical bud. Growth can resume from axillary buds if the apical bud is destroyed. In a few of the legume species, such as the pea, the hypocotyl does not elongate. Instead, it has hypogeal emergence, in which the cotyledons remain where planted. Additionally, the epicotyl elongates and pushes the growing point above the soil surface, effecting emergence. Remember, most legumes have epigeal emergence, in which the cotyledons are pulled above the soil surface during germination. However, grasses, such as corn and small grains, and certain legumes, have hypogeal germination, in which the cotyledons remain below the soil surface. You should next study the section on plant leaves in your text. Note that you should learn the leaf parts of broadleaf plants and grasses. You should also learn the functions of the parts. The first section for study is leaf anatomy. You should now stop the VCR, study the section on leaf anatomy in your text, and then return to the VCR for further tutoring. As you studied this section on leaf anatomy, you should have noted that leaves arise from nodes on the main stem or branches. The major functions of leaves are photosynthesis and gas exchange with the atmosphere. Leaves may be classified as being parallel veined or netted veined. This grass leaf has parallel veins, in which the veins run parallel throughout the length of the leaf. This legume leaf has netted veins, in which the veins have multiple branching, forming a lateral network. Note the internal structure differences between the leaves of dicotyledonous or dicot plants and monocotyledonous or monocot plants. Both dicots and monocots have a layer of epidermal cells acting as protection for the other cells of the leaf. The epidermis is covered with a cuticle, or wax-like material. In addition to providing protection for the epidermis, the cuticle helps maintain an adequate amount of internal water in the leaf by preventing the evaporation of water. Both dicot and monocot leaves have mesophyll cells, in which photosynthesis takes place. But only dicots have palisade cells, arranged side by side near the top leaf surface to catch the sunlight. The dicot leaf's spongy parenchyma cells have large pores between them, allowing for gas transport throughout the leaf. The translocation system, or transport system, in monocot and dicot leaves is made up of the xylem and phloem. Water and minerals are translocated from the plant roots through the stem and throughout the leaves in the xylem. Sugars and other materials are transported from the leaves to other plant parts through the phloem. The stoma are openings in the epidermis of the leaves and are surrounded by guard cells. Guard cells regulate the opening and closing of the stoma, which control the rate of gas exchange with the atmosphere. With the ability for gas exchange with the air, ready translocation of water and other materials, and the efficient arrangement of cells containing chloroplasts, the leaf has a very efficient arrangement for its principal function of photosynthesis. The next section on leaf morphology will discuss the external parts of leaves. It is important that you learn these parts because they are essential in properly identifying crops and weed species. You should now stop the VCR, study the section on leaf morphology in your text, and then come back to the VCR for further tutoring. Leaf morphology refers to the outward appearance and structure of crop leaves. Leaf morphology is especially useful in identifying species of plants. In subsequent units, you will need to know how to recognize differences in the various leaf parts in order to identify many of the important plant species. Some plant leaves are pubescent. This means they have hair-like structures on the surface. Other plant leaves are glabrous, or without pubescence. Pubescence may protect leaves against small insects and may also slow the loss of water from the leaf. Note in the figure labeled parts of a grass leaf in your text that a grass leaf consists of a sheath, collar, and blade. The sheath is fastened to the stem at a node and extends up, enclosing a section of the stem. The junction of the stem and blade is the collar region. Depending on the species or variety, the blade of grasses may be of different widths and lengths, or may be at different angles from the horizontal. Leaf collars may be continuous or divided, may vary in width or may be inconspicuous. Two different structures may be located on the inside of the collar region: the ligule and auricle. Ligules may vary in shape and size, and may be membranous or paper-like, hairy, or may not be present at all. Auricles may vary from long and claw-like to short to absent. The arrangement of leaves in the leaf buds, or leaf vernation, may be folded or rolled. Folded vernation usually results in a flattened leaf bud, and rolled vernation in a more cylindrical leaf bud. Legume leaves may be unifoliolate, having one leaflet or blade per leaf. The unifoliolate leaflet is usually attached to the stem or branch by a petiole. Legume leaves may also be trifoliolate, having 3 leaflets. Here, a petiolule fastens the leaflet to the petiole. The petiole then attaches to the stem or branch. Some plants have multiple leaflets with more than 3 leaflets per leaf. Some leaves terminate with a tendril, a viney-like structure. Stipules may be found where the petiole attaches to the main stem. Different plant species have stipules that vary in shape and size. They may range from prominent, narrow, to hair-like. Let's now study the sections on functions of stems and stem anatomy. The primary objectives in this section are to identify the anatomical parts of the stem and to describe the functions of the stem parts. You should now stop the VCR, study the sections on stem functions and stem anatomy in your text, and then return to the VCR for further tutoring. Stems perform some very important functions. These functions include translocation of water and mineral solutes from the roots to other plant parts, translocation of sugars and other synthesized food from the leaves to other plant parts, support of other plant organs, all above ground growth, including leaves, floral structures, and branches, originate from buds produced at the stem nodes. Stems may function as storage organs, as in sugarcane. Stems that contain chlorophyll manufacture some carbohydrates. Stems may also, in some species, be used to propagate new plants. The diagram of a longitudinal section of a dicot stem in your text shows the internal structures of the stem. The location of the primary phloem outside of the xylem is a common characteristic of both dicots and monocots. Synthesized food is conducted from the leaves to other plant parts, chiefly through the living cells of the phloem. The xylem vessels are non-living cells. Water and mineral solutes are conducted from the roots to other parts of the plant through the xylem. The epidermis is made up of cells that protect the inner cells of the stem. The pith cells, found in non-hollow stems, provide storage of food materials in the plant. An important point to note about a dicot stem is that it has a vascular cambium layer that forms a ring within the stem, with phloem on the outside and xylem on the inside. This cambium layer gives rise to new phloem and xylem, and therefore the legume stems can increase in stem diameter by cell division. In monocot stems, however, there is no cambium, and therefore any increase in diameter of the stems results from cell enlargement. Note again that the phloem is to the outside, and the xylem is to the inside of the stem. There are 2 types of monocot stems. One is called a stalk, which has vascular bundles scattered throughout the ground parenchyma of the stem. The other stem type is a culm, in which vascular bundles are scattered in a ring of ground parenchyma. The center of a culm stem is hollow. Crops with culm-type stems are often used as bedding material for livestock, providing the animal with a light, fluffy resting place. The next section for study is the modified stems. We usually think of stems as being somewhat upright or vertical, performing the functions discussed in the previous unit. Some crop species have modified stems that perform the functions of vegetative propagation and spreading the area of a single plant. You should now stop the VCR, study the section on modified stems in your text, and then return to the VCR for further tutoring. Four kinds of modified stems are illustrated in your text. A stolon is a horizontal stem that grows above the soil surface. New shoots can form from each node, and each of these shoots can produce a new plant. Rhizomes are horizontal stems that grow below the soil surface. New shoots or plants can also form from the nodes on the rhizomes. Stoloniferous and rhizomatous plants are usually considered to be sod-forming in nature. Sod-forming species create a carpet-like stand and are often grown in areas such as grass waterways to reduce the amount of soil erosion. A third kind of modified stem is a tuber. The most common example is a potato. Note that there is the possibility of a new shoot developing from each node or eye on the potato. The haplocorm, as found in Timothy, is the fourth example of a modified stem. After Timothy has been cut for hay, or after a long winter, carbohydrates from the haplocorm are used to furnish energy for regrowth from the buds at the crown of the plant. The next section for study is entitled Plant Roots. The objectives of this section are to identify the various parts of plant roots and to describe the functions of the parts. Roots anchor the plant in the soil, absorb minerals and water from the soil, and transport water and minerals to the plant stem, where it will be transported to other plant parts through the xylem. You should now stop the VCR, study the section on plant roots in your text, and then return to the VCR for further tutoring. Plant roots. Let's review some important points. Root hairs are single-celled extensions of the root epidermis and develop primarily in the zone of maturation. Root hairs form new root growth. Most of the absorption of water and minerals is in this area of new root growth. New root growth occurs primarily in moist, well-aerated soil. Let's now study the development of the total root systems of crop plants. Note that many legumes and other dicotyledonous plants have taproot systems that may be 3 to 5 feet or more in length. An alfalfa plant that is several years old may have a taproot that is 10 to 30 feet deep, depending on soil type and other factors. The root system of an annual legume, such as soybeans, may be only about 5 feet deep and branched to the extent that it appears almost fibrous. Grass roots are said to be fibrous because instead of having a main root, they form a network of roots from the crown area of the plant. The depth of grass roots ranges from about 2 feet for some sod-forming species to more than 5 feet for large grass plants such as corn. The fibrous root system of grasses makes them good species to grow as cover crops for erosion control. The last section for study is the section on grass and legume inflorescence. The word inflorescence refers to the flowering parts of the plant. You should learn the parts and types of grass and legume inflorescences and how they vary among crops. The information in this section is important and necessary to the understanding of yield, plant breeding practices, and plant identification. You should now stop the VCR, Study the introductory remarks and the parts of a grass floret in your text, and then return to the VCR for further tutoring. The first topic of review under this section will be parts of a grass floret. The grass floret, or grass flower, can be defined as the lemma and palea and everything that lies in between. A typical young grass floret will have a lemma, palea, and male or female reproductive structures. An older mature floret, however, will have a lemma, palea, and kernel. The grass floret is normally perfect, which means that it has both male and female parts in the same flower. The male part, called the stamen, is made up of anthers and filaments. The female part, called the pistil, is made up of the stigma, style, and ovary. Therefore, you should logically guess that it would be self-pollinated, unless some additional mechanism exists to prevent it. While the grass floret is considered perfect due to its reproductive components, it is also incomplete because it is missing the corolla, or petals. It's important to remember that the fertilized ovule and the ovary develop into the caryopsis, or kernel. The male parts of the floret have usually disintegrated by the time the caryopsis matures. Corn is an example of a monoecious plant, meaning that the plant has imperfect flowers, but both male and female flowers are on the same plant. The corn tassel contains male flowers, and the ear shoot has female flowers. A dioecious plant also has imperfect flowers, but male and female flowers are located on separate plants. The next topic for study is the grass spikelet. The parts of the spikelet are frequently used in identifying plant species or in discussing components that determine seed yield. You should now stop the VCR, study this section in your text, and then return to the VCR for further tutoring. The picture and diagram of the wheat spikelet in your text may be used to study the various parts of a grass spikelet. Note that the spikelet consists of the two glooms and everything in between. In wheat, the glooms enclose 3 or more florets attached to a central stem, called the racula. Note that the number of developed florets per spikelet varies with the species. For instance, oats generally have 2 florets per spikelet, while orchard grass generally has many developed florets per spikelet. Developed spikelets, or kernels per spikelets, also may be influenced by environmental factors such as weather and soil fertility. The next topic for study is parts of a legume flower. Study this section in your text, and then come back to the VCR for further tutoring. The figure in your text shows a closed and open legume flower. When the bee visits a closed flower during its search for nectar, the keel is tripped, exposing the staminate column, and thus opening the flower. This tripping action dusts the bee with pollen, and when the bee visits another flower, cross-pollination can occur. Let's now dissect a legume flower. Note the visible parts of this flower from a sweet pea. The calyx is made up of 5 fused sepals. The corolla is made up of the standard petal, 2 wing petals, and 2 fused petals that form the keel petal. When the standard petal is removed, followed by the wing petal, and then the keel petal, the reproductive parts are exposed. The stamen is made up of anthers attached to filaments. There are 9 filaments fused to form the staminate column, and 1 filament that is free. The removal of the anthers, filaments, and staminate column exposes the stigma, style, and ovary of the floret. The splitting of the ovary exposes the ovules that are ready to be fertilized. The anthers produce pollen grains that contain the reproductive cells. The sweet pea flower is a typical legume flower, but it is several times larger than the flowers of normal crop legumes, such as the soybean and alfalfa. The peanut flower is a typical legume flower, with one exception. After pollination and fertilization, the flower develops a gynophore, or peg, which elongates and enters the soil. This gynophore produces a pod-containing seed. The next section for study is inflorescence types. Note that we are only going to discuss 4 basic types of inflorescences commonly found in agricultural crops. You should now stop the VCR, study this section in your text, and then return to the VCR for further tutoring. In the spike inflorescence, the spikelets are directly attached or sessile to the central axis of the inflorescence. This central axis is called the rachis. Note that the point of attachment of each spikelet is a node on the rachis. Rye and barley spikelets differ somewhat from wheat, but their inflorescence type is also a spike. Note that the 6-rowed barley spike has 1 floret per spikelet and 3 spikelets per rachis node, giving an appearance of 6 rows of florets per spike. The 2-rowed barley has 1 floret per spikelet and only 1 fertile spikelet per rachis node, giving an appearance of 2 rows of florets per spike. The second type of inflorescence we are studying is called raceme inflorescence. Several legume species, such as sweet clover, fall into this category. Unlike spike inflorescence, raceme flowers are not attached directly to the rachis. Each raceme floret is attached to a pedicel. It is this pedicel that then attaches to the rachis. The third type of inflorescence is the panicle type. The spikelet usually contains 2 florets, the primary and the secondary. Spikelets may contain 1 to several florets, depending on the species and environment. In a panicle, the spikelet is attached to a pedicel that is attached to the panicle branch. It is the panicle branch that is then attached to the rachis of the inflorescence. The final type of inflorescence we will study is called head inflorescence. 2 examples would be red clover and sunflower. These 2 heads look different, but they have something in common. In the red clover head, the flowers are clustered and attached to an enlarged, shortened receptacle. In the sunflower head, there are ray flowers and disc flowers that are also attached to an enlarged, shortened receptacle. Be sure to carefully study all of the information presented in the text and in other sources. After you have completed your study, you should be able to take the self-evaluation test at the end of the chapter in your text. This is the end of the study of crop plant anatomy. Please rewind this tape before returning it to the lab monitor.